A very pleasant good morning to everyone. Myself Jayapiriya US, pursuing my PhD doctoral degree under the supervision of Professor Sanket Govil, Head of MMNE Lab, Department of Electrical and Electronics Engineering, BITS Pilani, Hyderabad campus. I am here to present on the topic High Performance Machine Supported Gold Nanoparticles Based 3D Printed Anode for Non-Enzymatic Biofuel Cell. We would like to thank the financial assistance provided by IEEE NTC Student Earnest Setup Program for attending this IEEE NMDC 2021 conference. Before I could start, I would like to tell about the campus which I have come from, which is one of the campuses of BITS Pilani University. This campus was established in the year 2008. This work concentrates on additive manufacturing, that is also called as 3D printing, for development of energy storage and energy harvesting applications. There are different types of 3D printing techniques such as DLW, FDM, SLA, IJP, in which FDM, that is fused deposition modeling, has gained a huge attraction in this energy field. Different types of uh, parts of the fuel cells or batteries can be made with the help of 3D printing technique, which has been explored which has been explored very highly. In this work, we will also be talking about non-enzymatic biofuel cells. Non-enzymatic biofuel cells are those which uses biofuels for harvesting power without the presence of enzymes. Certain amount of work has been done in this upcoming area. We will be seeing some of the important works in this field. Zaho et al. Has developed a nanoporous gold anode and a copper complex cathode as a cathode for a non-enzymatic biofuel cell. They have found that it was able to harvest a power of 100 microwatts per centimeter square with a stability for around 30 days. The next work was done by UE et al., where they have fabricated a highly porous platinum nickel nanoalloys using a simple electrode deposition technique, which was capable of harvesting a power of around 7 microwatts per centimeter square with a stability for 90 days. The next work which has been done at our group is a microfluidic non-enzymatic biofuel cell, wherein we have used a gold electrode deposited anode and a copper electrode deposited cathode on CNT Baki paper, which was integrated on a paper-based Y-shaped microfluidic channel to to harvest power. This biofuel cell was capable of producing a power of 12 microwatts per centimeter square, which is quite commendable for a paper-based platform. These are some of the other works where enzymatic biofuel cells has been developed at our group using different electrode materials like Baki paper, pencil graphite, laser induced graphene. These electrodes were capable of producing a good amount of power density also. This work we will be talking about 3D printing and the fuse with electrode deposition of 2D materials and gold nanoparticles. First, commercially available graphene filament was used for making 3D printed electrode with required dimensions. The next step was to electrode deposition of Mexine. Mexine has been attaining a very huge attraction because of its high electrical conductivity with an added advantage of high hydrophilic surfaces. After the electrode deposition of Mexine, gold nanoparticles were also electrode deposited over these Mexine coated 3D printed electrodes. To confirm the efficient electrode deposition on the 3D printed electrodes, scanning electron microscopy was done. It can be seen the multi-layered Mexine has been uniformly deposited over the 3D printed electrode by restoring its layered structure. Over this, the electrode deposition of gold nanoparticles has been successful, which is implied from these figures where gold nanoparticles are uniformly distributed over the Mexine coated 3D printed electrodes. Further, to understand electrocatalytic analysis were done to optimize the parameters for electrode deposition and the catalytic activities of the glucose biofuel cell. It was found that at an electrode potential at an applied potential of 0.18 volts, for 15 minutes of electrode deposition time gave a better current output, followed by the number of cycles required for electrode deposition of nanoparticles, gold nanoparticles using cyclic voltammetry was also optimized. It was found that as the number of cycles for gold deposition increased, the gold loading was also increased, whereas after a particular number of cycles, that is 10, the even though the gold nanoparticles loading increased, it gave a lesser current output. This was because when the number of layers of gold kept on increasing, the size of the particles also increased. Hence, the number of cycles was decided to be 10 and has been used for the further experiments. Also, the glucose concentration in which the bioanode has to be performing was also optimized using linear voltammetry in a concentration range of 10 to 60 millimolar. It was found that 40 millimolar given good performance and was used in the experiments. The bioanode characterization was done using a cyclic voltammetry in the potential range of -0.3 to 0.9 volts with a reference electrode of AgCl saturated in KCl. It is clearly understood that in the presence of mexene AU on the 3D printed electrode, a well-defined redox peaks were observed corresponding to the glucose oxidation. Similarly, to understand the interface interface effect of mexene and AU on the 3D printed electrode, electrochemical impedance spectroscopy was done. It can be clearly seen in the presence of mexene and AU gold nanoparticles on the 3D printed electrode. It has a lesser linear area and charge transfer resistance. Thus, these electrodes prove to be a good anode for a non-enzymatic biofuel cell. Hence, to understand and to confirm this, we have done the polarization studies using a chrono, and the plots were done with the help of chronoamperometry. It can be seen that in the presence of a mexene AU coated 3D printed electrode, the biofuel cell was capable of producing a high power of 18 microwatts per centimeter square. Also, the electrodes were able to give around 90% of its initial power performance until 10 days. Hence, we conclude saying that commercially available graphene filament was used as a bioelectrode material for enzymatic biofuel cell with the help of a FDM 3D printing technique. Gold nanoparticles were used as an electron transfer enhancer, and 2D material mexene was used as a supporting material for deposition of gold nanoparticles. A model biofuel setup was done which gave a power of 18.2 microwatts per centimeter square with an open circuit voltage of 360 millivolts. Hence, 3D printed mexene supported bioelectrodes could be of great potential in the energy technologies of the future. In our lab, we do have a micro and nano fabrication techniques such as photolithography, CO2 laser, 3D printing, soft lithography and paper-based device fabrication techniques, along with characterization facilities which would be helpful in material synthesis. Also, in our college, we do have a class 100 and class 1000 clean room. This is our group MMNE lab at BITS Pilani, Hyderabad campus. I would like to thank everyone for this wonderful opportunity to showcase our work on a hybrid device of 3D printing and non-enzymatic biofuel cell. Thank you.